Today, we're embarking on what I like to call a botanical rescue mission. We're going to dive into the incredible science behind saving one of the world's most exotic and most endangered flowers from the brink of extinction. Let's get started. So this is the big question, right? It's really the heart of this whole story. When a species is right on the edge, can science actually step in and pull it back? Well, we're about to find out. Our mission begins with the star of the show, a flower of incredible, mysterious beauty that's now facing a very, very real threat. Just look at this thing. This is the Black Orchid. It's from the island of Borneo, and its huge green flowers and that deep, almost painted-on black tongue make it completely unique. It's a true masterpiece of nature. This right here is kind of the perfect storm of factors that makes this orchid so vulnerable. First, it's endemic, which means you can't find it anywhere else on Earth. Its beauty gives it a high economic value, but, and this is a big but, it only flowers a couple of times a year. All of this together means it's critically endangered and incredibly hard to find in the wild, which means we're running out of time. So how in the world do you save a plant from extinction? Well, the answer lies in what you could call a genetic rescue mission. The key strategy is a concept called hybridization, and in simple terms, you can think of it like botanical matchmaking. Scientists cross two different but related species to create a new hybrid. The goal isn't just a new flower, it's about strengthening the gene pool and building resilience. And the mission objective was crystal clear. Use hybridization to pull the Black Orchid back from the brink of genetic extinction, to create a new generation that could carry on its legacy. But hybridization isn't random. You can't just pick any two flowers. The success of this entire mission depended on finding the absolute perfect partner for our Black Orchid. And here they are. On the left are endangered Black Orchid, Coelogyne Pandurata, and on the right, its chosen partner, Coelogyne rumphii. They look pretty different, don't they? Let's break down why this specific pairing was chosen. And this just brilliantly illustrates the strategy. You've got the Black Orchid with its big, dramatic, but very rare blooms. Then you have Coelogyne rumphii with smaller, less dramatic flowers, but, and this is the key, it blooms almost constantly. The hope was to combine the beauty of the Black Orchid with the flowering frequency of its partner. So, with hundreds of orchid species out there, why this one specifically? The answer, as it so often does, comes down to genetics. This number is the key to everything. A previous study revealed that these two species share an 87% morphological similarity. In the world of plant breeding, that is an incredibly strong indicator of compatibility. The more closely related they are, the higher the chance of a successful cross and, hopefully, fertile offspring. They were a near-perfect match. So with the couple found, the delicate work began. In a controlled lab environment, scientists painstakingly transferred pollen by hand. Once the fruit formed, they grew the tiny seeds in a nutrient-rich gel, a process known as in vitro or in glass culture, to give them the best possible start in life. So, did it work? This is where the story gets really, really interesting. The results were not what anyone expected. 100%. That was the initial success rate. Think about that. Every single attempt to cross these orchids resulted in a fruit. This included the standard cross, a reciprocal cross where the parents' roles were reversed, and even self-pollination. This was a phenomenal start. However, it wasn't all smooth sailing. The team quickly ran into their first hurdle. As this chart shows, the standard crossing method, where the Black Orchid was the female parent, had a 50% fruit drop rate. Half of the potential seeds were lost before they even had a chance to mature. The next piece of the puzzle was time. How long did it take the fruits to mature and be ready for harvest? Here, the crossing method was actually the fastest of the hybrids, taking 158 days. The reciprocal cross, where the parent roles were swapped, took over a month longer. But this was the breakthrough. The most critical factor for success is germination time, how fast the seeds sprout into what's called a protocorm, which is the very earliest stage of a new orchid. And just look at that first bar. Using the standard crossing method, seeds began to germinate in just 22 days. That is incredibly fast, more than twice as fast as the other methods. This was the game changer. So let's put it all together. This summary table tells the whole story. Even though the standard crossing method lost more fruit at the beginning, its lightning-fast germination time was the deciding factor. The takeaway was clear. Despite some early losses, this method offered the quickest, most efficient pathway to creating viable new plants. And with that data, the outcome of the mission became clear. This wasn't just a successful experiment, it was a new beginning. The conclusion from the study itself really says it all. A 100% success rate in forming fruit and a germination time of just 22 days. This is a recipe for success, a repeatable method for propagating this endangered species.
And this is why this matters so much. Beyond just this one beautiful orchid, this whole process, from identifying a genetic partner to analyzing the trade-offs in breeding methods, serves as a powerful blueprint. It's a strategy that can be adapted to help preserve the genetic diversity of other threatened species all around the world.
Thanks to this painstaking scientific work, the future for the black orchid is no longer one of extinction, but one of hope and regeneration. A new future is quite literally blooming. It leaves us with a powerful thought. By combining careful observation, genetic science, and a whole lot of patience, we can intervene and protect life. If we can write a new future for one endangered orchid, what other species can we save? What else is possible?